Alrighty, let's talk about mutations. So, there are two origins of mutations. You can either inherit them, meaning that the original mutation came from either mom or dad, and when the two cells combined to make you were born with that mutation, or you can acquire mutations throughout your lifetime in your own body. Now, there are a bunch of different causes of mutations. It can be from environmental factors like poor diet, smoking, radiation, mutagens, radiation, like I just said. Non-disjunction. Non-disjunction is when chromosomes do not separate properly during meiosis or mitosis. That's how Down syndrome works, for example. You end up with an extra copy of chromosome 23, or you could be missing a chromosome altogether. And then certain diseases and viruses can also cause mutations in your body. And there are two locations that mutations can be. You have what's called autosomal mutations. These are the ones that occur in your everyday body cells that you have. So like, for example, maybe there's a skin cell on my hand somewhere that has a mutation in it. And those ones are not really dangerous unless they are cancer, because if one single cell on my hand skin is mutated, that is not going to affect anything else in my body, okay? And when that cell dies, the mutation is going to die with it, so it will not be very important or dangerous unless, of course, it is cancer, and we'll talk more about cancer in a minute. The sex-linked ones are the ones that can be a little bit more tricky. So again, these are the ones that occur on sex chromosomes during the meiosis process that can be passed down to the offspring. Now, with this one, that since you begin with literally that one cell that has a mutation in it, every copy of that cell makes will also have the mutation. So that mutation will be in every single cell in your entire body, and you will live with that for your entire life. Now, again, not all mutations are bad, but depending on the mutation, it could be bad, could be fine, could be not important at all whatsoever. All right. Now, we know that mutations are caused by actual errors on the DNA strands or the RNA strands themselves. So here's how that works. There's 4 types. You can have a deletion, which is where one base is deleted, so ATC could turn into AC. You can have substitution, where one pair is exchanged for another one. Like in this one, you see that T has turned into C. And then you can have an addition, where another one is added. And then, like I said, with chromosomal, you can have chromosomes be excluded. You could have one added, swapped, any combination of that. And like I've said, sometimes they're good, sometimes they're bad. Totally depends on what exactly the mutation is. All right. Now, cancer is one of those ones that is definitely bad in most cases. So with cancer, the mutation causes your cells to not stop growing. So in the cell cycle, there are checkpoints that the cell has to go through. And normally at a point when a cell gets too big or it's reached a certain age, then it will stop reproducing, stop growing, and kind of go dormant. Cancer is when cells do not go dormant ever. They will keep reproducing until you end up with tumors. Some cancers can metastasize, meaning they can break off in pieces and travel throughout your body and create other spots of cancer throughout you. But know that it is caused by uncontrolled cell division and is not generally good. Now, let's talk about how to actually translate and code DNA. So let's say that I have a strand of DNA and I'm in my cell and I am trying to copy it. So RNA is how you copy DNA. So in this picture here, you can see that this DNA molecule, the one in blue, part of it has been unzipped, and this red thing, which is the RNA, has come in and it is making a copy. So we know that with normal DNA, A pairs with T and vice versa, and C pairs with G and vice versa. And we can see that they literally fit together. Now, when RNA comes in, we see there's a little bit of a difference. Instead of T, there is U. So with RNA, A and U pair together, G and C still pair together, and there is just no thymine at all in RNA. So those are your DNA rules and your RNA rules. All right. Now, once the RNA comes in and makes the copy of the DNA strand, this strand here will go to a thing called a ribosome in your cell, and that ribosome is what builds together the actual amino acid chain that will eventually become a protein to help your body function. All right, so let's talk about actually translating. We've set our base pairs already. This is what they look like. You can see that they pair together because cytosine has one little hexagon, guanine has two, so they fit together. Adenine has two, uracil has one. Same thing with thymine. So they all fit together literally like a puzzle. All right, so now let's talk about codons. We know that every 3 rungs of the ladder equals 1 codon, and 1 codon equals 1 amino acid. So how do you figure out what amino acid or what codon means what amino acid? You use a codon wheel. So let's say we have the codon AUG. Fun fact, that's normally what your genes will start with. We will go to our codon wheel, which looks like this, and if our codon is AUG, we will first go to A in the center here, and then we will go to find our second letter, which is U, and then we'll find our third letter, which is G, and we end up with this amino acid called methionine, symbolized by MET, and we see that there's a start next to it. And methionine usually means, like, it tells the RNA strand to start coding here, so that's called the start codon. So AUG basically means start, or MET. Now let's add another one. Let's do GGG, and let's go see what GGG codes for. All right, so again, start in the middle, find G, find the other G, find the other G. That codes for something called glycine. So that would be glycine. And it goes on and on and on like that. And you'll notice in the codon wheel too, you have 1 codon for start, meaning like start coding here, and you have several stop codons. So once the mRNA strand reaches the stop codon for in that particular gene, it will stop coding, cut itself off so it doesn't mess up the protein and add extra things that it doesn't need in there. All right. Now, important thing to remember, it is not the DNA strand that goes to the codon wheel, it is the RNA strand. You are coding from the RNA strand. So if I give you a DNA strand, you have to translate that into RNA first, and then use the RNA strand to go to your codon wheel and figure out what your amino acids are going to be. So that worksheet on your desk, those are the answer keys for it. You can go back and check those once you are done, but use your codon wheel, translate your original DNA sequence into your mRNA, and then figure out what amino acids are going to make it up. And then throughout it, you'll be able to see how exactly the different types of mutations mess up the amino acids that go into the protein.